Good morning, everyone. Welcome, everybody, to the second to last talk of the 2025 Royal Tyrrell Speaker Series, sponsored by the Royal Tyrrell Cooperating Society. For those of you who don't know me, I'm Ilaria Paparella, and I'm one of the curators here at the Royal Tyrrell Museum. I'll be your host for today. So this morning, we're pleased to welcome Dr. Yan-Yin Wang. Yan-Yin came from Canton, China. He started his undergraduate in geology, but his interest gradually shifted to paleontology and eventually evolutionary biology. This led to a Master of Science degree program at the Institute of Vertebrate Paleontology and Paleoanthropology in Beijing. After that, he moved to Canada for a PhD program at the University of Alberta in Edmonton under the supervision of Dr. Colville Sullivan. During his PhD, Yan-Yin combined his interest in 3D modeling and more traditional paleontological methods to explore patterns in evolutionary transitions of dinosaurs and their next of kin. For his PhD thesis, he focused on the composition of rib cages in some dinosaurs and their close relatives, to then infer on how the various rib cage elements may contribute to breathing. So today, Yan-Yin will present a talk titled Anatomy and Biomechanics of the Ribcage in Birds, Crocodiles, and Dinosaurs, with the implications on the evolution of ventilation. So without further delay, it is with great pleasure that I present you Dr. Yan-Yin Wang, and the floor is yours.
Okay, thank you for the kind introduction. You are upselling me maybe a bit too much. I will try to make this talk entertaining and educational. So let's begin by setting the stage a little bit. So breathing is more complex biological processes that involve many technical terms. Sometimes we call respiration, sometimes we call ventilation. So if we follow Perry and his colleague, then we can classify the entire breathing process into three stages. So you have the neural control, where our brain will send out information to the muscles around our body as well as the organ, so that we can control the pumping of air, carbon dioxide, and oxygen into and out of the respiratory organs, such as our lungs. So this would be the active pumping part of the breathing process. And in a more technical term, then we call them ventilation. So my talk today will focus mostly on the active pumping part. So we're taking a more mechanical as well as anatomical approach. And this is also one of the venues that we can really look into the evolutionary history regarding breathings. Right. So my study group is dinosaurs and their next of kin. In a more technical definition, this group includes, this group is called archosaurs, the dominant lizards back in the Mesozoic time. Cooler name compared to dinosaurs, in my opinion. So even in scientific literature, we can define the entire group Archosauria as the last common ancestor shared by the house sparrow on the left and the Nile crocodile to the right, and as well as all of the descendants. So this will include all the modern birds, all the modern crocodilians, and their ancestral, what we call the pseudo-Suchian, pseudo meaning fake, so like a fake crocodilians in the past, as well as the more famous non-avian dinosaurs, like this silhouette based on the UALVP2 specimen from the University of Alberta. So this is a big group for us to study, and we only managed to study a selected text among the entire group. So we are cherry picking in some sense for this presentation. So we have a lot of diversity across the entire Archosauria lineage. However, if we look at breathing, they almost always use a method called the costal ventilation. We have this same mechanism as well. So if you're happy, you can feel your collarbone and push your finger a bit downward and try to breathe in as hard as you could. You can feel your chest is pushing upward. So that's the effect of the costal ventilation. Costal meaning ribs. So it's a way to pump air into and out of the body cavity using the movement of the rib, powered by the costal muscles, if you would. So how can we study ventilations in the context of evolution? So we can look at the anatomic or the anatomy of the ribcage by answering the questions like what makes up the system. So this is the system for breathing includes the ribcage, includes the lung tissues, includes muscles, and we can use anatomical approach to answer this question on the left. And then based on the anatomy of the rib cages, then we can answer questions as to how this system might function in selected species, and then further down the road, we can compare the performance of system across multiple species. Like, do we have any species of dinosaur that can breathe better compared to other kins, for example? So for the first part of my talk, we will focus more on the anatomical side on the left, and then we'll move on to the 3D modeling and answer the question how the system might function and how we can explore explore ventilation after my project is finished. Okay, so what makes up the system? The first question we need to answer is really not to answer this bigger question. We also need to answer a smaller question as to where does the ribcage start. Right? In mammals, we always have 7 neck vertebrae. So we can feel the back of our neck. If you push your finger further down close to your shoulder blade, you will see a bony bump at the back of your neck. That is either the end of your neck or the beginning of your chest. However, in archosaurs, the question is more complex. So they have what we call indeterminate number of neck vertebrae. So in some archosaurs, like the American alligator over here, it's only 2 weeks old, but you can see it has about 9 neck vertebrae. So it's a decent number. But if we look at modern birds, like the dodo over here, they have way more than 10 neck vertebrae. So we don't really have a clear number of neck vertebrae in which we can start from the back of the neck, start from the back of the head, and then count, say 9 or 10, and then say that's the end of the neck. We don't have this option. Now, in early days, then Alfred Romer, the famous paleontologist, actually defined the transitions, like the junction between the neck and the chest, using what we call the first connection between the vertebral column and the chest bone, called the sternum, by various rib segments. So over here, we can see some illustration, some figures where we can start from the back of the vertebral column, going down to the what we call the vertebral rib, and then further down you have the blue color, so those are the sternal rib made up of cartilage, and then further down on the belly side, then we have the sternum. So we can find the first connection and say that is the beginning of the chest. All works quite well in the eyes of Romers, and in my opinion, this is a really good definition to follow, really precise and actionable. However, in many dinosaurs, these and their next of kins, the sternal ribs are cartilaginous, and cartilage fossilized not so well compared to ossified bones. Right? So in many cases, you don't have the sternal ribs preserved in the fossil record. You don't have the complete sternum preserved in the fossil record. To make it worse, many of the fossils come from what we call bone beds. So you have disarticulated bone elements scattered across the entire area. So we don't really have articulate skeletons all the time. So we as paleontologists then will have to deal with individual vertebrae laid out on the table in front of us, and we have to make the decision. What vertebrae are we looking at? Are they from the neck or are they from the chest? So we will. And based on this challenge, then we say maybe we can look at the morphological features, like the anatomy of the backbone vertebrae, and then see what kind of features are changing from the front end of the neck all the way to the back end of the chest. So there might be some pattern we can extract from the anatomy. So in order to do that, we look at a selected number of features. We look at 8 features overall, looking at the unambiguous neck vertebrae all the way to the unambiguously chest vertebrae. Right? So if any pattern exists, then we capture them, we encode them into discrete characters like 0, 1, 2, and 3, for example. And 4 characters out of all of these demonstrate a somewhat consistent pattern. This is the exciting part. So if we look at the vertebrae in dinosaur and next of kin, like on panel A over here, you can see there's a bump or protrusion we call the parapophysis. And as we move further towards the tail, the location of the parapophysis is shifting towards the back side of the vertebrae. So they're moving further towards the back of the animal. And another feature is the diapophysis over here. So it's more or less the almost equivalent to the transverse process. It's a little bit more dorsal, closer to the back compared to the parapophysis. And then we realize as the vertebra move towards the tail side, then the orientation of the diapophysis actually shifted from a more belly pointing direction to a more side pointing direction laterally. And we can also find other features such as the foramen magnum, where it's a location to house certain blood vessels running parallel to the vertebral columns, as well as what we call the hypopophysis, where it's the attachment site for some of the neck muscles. So 4 of these features are quite consistent, and we can use these features and then combine these sets of features to delineate. Basically, we have an entire vertebral column in front of us, then we can see which characters transition first, and then we say this is one landmark, and then which the second character will transition later, then we say that's a second landmark. In effect, we are classifying the entire vertebral column, such as the ostrich on the panel on the right-hand side, the top row. We can classify all these vertebrae based on the combination of 2 transitional features. Right? So over here, what we also realize is that the diapophysis orientation always takes place first, way before all the other features, and also before the establishment of the first connection with the sternum. So our definition can therefore house a region within the vertebral column where the first connection to the sternum might be located. Right? So we are not as precise as Romer's definition, but we can still delineate a small region of the backbone and say the ribcage likely start at that location. Maybe off by 1, off by 2. And so, in effect, we're actually revising what Mivart established back in 1877. If I remember correctly, Mivart was in the same time as Darwin, and also not quite in support of Darwin's idea at the time. So we are revising Mivart's idea and classify the entire vertebral column not into just neck and chest, but rather into neck and what we call a cervical dorsal region, and then the chest regions. So over here we have a diagram showing you how it works. Right? So over here we have the diapophysis orientation takes place first. So anything before this, we call them unambiguously neck vertebrates. And then we have the second character take place later. Then in this case, is the parapophysis on panel A, right? And then we can say anything after this, they are unambiguously dorsal vertebrate or chest vertebrate. And somewhere in between, labeled by the green color, so those are maybe vertebrate. So they could be neck vertebrate, they could be chest vertebrate, and we just call this region the cervical dorsal region. Maybe with more studies, we might be able to have a more precise delineations for the vertebral columns. And if you want to discuss more at the end, we can talk about some methods using machine learning and try to classify them based on training methods. But we should focus more on ventilation for today's talk. So as you have lunch after the talk, maybe you can walk in the gallery and then look at some of the skeletons, like maybe some of the death-posing Gorgosaurus, see if you can identify which one is the neck, which one is the cervical dorsals, which ones are the chest vertebrates. And other than the vertebral column, we also have rib segments. And in archosaurs, very interestingly, they have a structure that we call the uncinate processes. Uncinate meaning small hook. So this is a process at the back of the rib that looks like a hook. So it's first discovered in avian or bird anatomy literatures, but up to this point, we realize uncinate processes can be found in 3 modern lineages. One is what we call the Tuatara sphenodon in New Zealand. So this is one specimen on the left-hand side with the blue arrow pointing at the cartilaginous uncinate processes. It's not really well preserved because Tuatara is quite rare outside of New Zealand. So if you have the opportunity to visit New Zealand, the actual complete version of the uncinate processes, they are a bit longer compared to what I'm showing here on the figure. And within Archosauria, the group that I study, then we can still find uncinate processes in both modern birds and modern crocodilians. In modern crocodilians, they appear as a cartilaginous tab. So the figure in the middle under the Cayman crocodiles, so this is one dorsal rib of the crocodilian, and then this irregular shaped tab pointed by the blue arrow, that is the cartilaginous uncinate processes. It looks irregular because cartilage get dehydrated and then deform. When they are hydrated, then it's more smooth and plate-like. And in the case of modern birds, most modern birds, with exception of tinamous and emus, they all have ossified version of the uncinate processes. So those are the figures under Rhea americana, as well as the ostrich animation on the right-hand side. So they are labeled by the red arrow over there, showing you that's the bony versions rather than cartilaginous versions. But what about the fossil records? Right? In the fossil records, we have some documentations regarding the ossified version of the uncinate processes in a group that we call the Panaraptorans. So Panaraptoran is a group that includes all modern birds as well as some non-avian theropods that are really close to modern birds. So one example is the dromaeosaurid Lyncaraptor over here. So this is part of the chest of the Lyncaraptor. We are looking at a specimen on the right-hand side. And this long bone on the top, that is the scapula on the top. And then further to our left-hand side, that is the series of the neural spine from the vertebral column. And all those long rod extending almost vertically, so those are the rib segments. And attaching to those rib segments, we have some long branches extending towards the scapular blade as well as the vertebral column. So those are the ossified version of the uncinate processes. And they appear to be longer than the ones in modern birds as well. And in pseudosuchians, the ancestral crocodilians or crocodilomorph, then we also have evidence of the triangular-looking cartilaginous uncinate processes. So if you look at the Arapisuchus on the top right-hand side, then you have the zoom-out area or zoom-out view of the entire specimen, and then you have the zoom-in view outlined by the blue rectangle showing you the small triangular uncinate processes attached to the end of the dorsal ribs. And in Ornithischian dinosaurs, in one group called the Thessalosauros, I believe they are somewhat basal, they are positioned in a somewhat basal location on the family tree regarding the entire Ornithischians. I might be wrong because this group has been actively studied by scholars. But they realized they can find this tab-like structure, really large tab-like structures also attached to the back of the ribs. Now, the scholars such as Boyd Iscoli, they call this intercostal plates. So whether these are uncinate processes or not, or they are homologs or not, we are not 100% certain. In my opinions, and I would believe these are the homologous structures of uncinate processes. You can have your own opinions. We need more studies to know more about the distribution of uncinate processes as a feature. Now the problem arrives when we look at this, the uncinate processes in the fossil. Okay, we have the ossified versions in Panaraptoran. Great, they ossify, and then they can be documented in the fossil record as well as other bones. No problem whatsoever. But in Ornithischians, if these are uncinate processes, in Pseudosuchians, they are cartilaginous, meaning that we don't have a really good method to sample uncinate processes in those groups. They are biased by what we call taphonomic factors, just because cartilages do not fossilize as well. So we thought maybe we can find some kind of indicators on the side of the rib that can tell us whether the ribs would bear an uncinate process or not. In fact, we are finding what we call an osteological correlate, and in here we just call it an indicators. In order to find these indicators, then we survey, I think, 6 birds and 6 crocodilians. With permission, we remove the uncinate processes from the rib, damaging the specimens in the process. And then we can use microscopy technique to visualize what the attachment sites look like. And therefore, we can use this as a baseline reference and then look at what fossil dinosaur ribs look like and whether they have the same characteristics for the attachment site or not. In order to establish this attachment site, we realized when we look at the specimen after removing the uncinate processes, we realize the attachment site is both texturized, meaning it's quite rough compared to the surrounding bone tissues, as well as it's usually somewhat concave. So if we look at the rib on the side view, then you can see there's a black line on panel D over here. It's running almost up and down. But if we remove that uncinate processes, it would cave in a little bit. So it's a dimple on the edge of the rib. And at this point, we were still not quite certain if we were on the right track. But during one trip to the American Museum of Natural History, we found one fossil crocodilian rib over here on panel M and panel N. So with the zoom-in view on panel N, we can see there's an almost identical structures, almost identical dip with texturized morphology similar to the modern version, to the uncinate scars in modern birds and modern crocodilians. So that gave us confidence to apply this indicator to survey uncinate scars in fossil dinosaurs and their next of kin. And maybe we can say that if we can find this indicator in some species of dinosaurs, that means they likely would bear uncinate processes when they were alive. And so we did. We only look at 19 taxa. So they can be known species like the Gorgosaurus here from the University of Alberta. So right around here on the top next to the label, you can see some rough structures. That's where the uncinate scar is located. And we can see a better view by looking at panel A and panel B. So you can see the surface is kind of rough, and then you can kind of also see close to the label PR, what we call the proximal ridges. Then you can see it's almost caved in a little bit with the textures. And by surveying multiple species, we also realize there are some variations in terms of uncinate scar morphologies. In addition to being concave and texturized, in some species, such as from panel I to panel G, then we have specimens from hadrosaurs, we have specimens from Centrosaurus, the horned dinosaurs. The uncinate scars on these groups, they look almost like an indent made by a toothpick, the size of the toothpick. So they're really tiny and small. And we got really lucky with one specimen here at Tyrrell Museum. So it's demonstrated on panel F and panel D. So we have a rough scarring tissues on top labeled by CT. So what we believe those might be calcified tissue that has been fossilized, and those might be the tissue binding the uncinate processes to the vertebral ribs when the Centrosaurus was alive. It was quite precious, so we did not do any destructive sampling at the time. And I know what you are thinking. Right? When I showed that to my supervisors and colleagues, then they were not very convinced. They were encouraging with a dose of skepticism. Right? We could have many other explanations for those quote-unquote uncinate scars. It could be biomarks, it could be muscle attachment, it could be other taphonomic factors, it could be errors by technicians. We never know, right? So we need more evidence. And so happened I knew a paleodontist at the time, and he was very enthusiastic about cutting up teeth and grinding them down such that they can see small tissues and even cells within the teeth. So he told me, why don't you cut the ribs and grind them down, make some histological sections. So as a barbarian, I think that's a great idea. So we got some ribs from turkeys, we got some ribs from caiman crocodile. We make what we call the histological sections that you can see those blue, those purple and red panels. Those are histological sections from the turkey on the left and from the caiman on the right. So let's orient ourselves a little bit. So on the left-hand side, you have histological section from a turkey. On panel A, there's an overview of what the section looks like. And on the bottom side, below the BD label, that's the section belong to the rib segments. And above the BD label, that's the section belong to the uncinate processes. So we are looking at how the tissues bind the uncinate processes to the vertebral ribs. And we can talk a lot of the tissue details. If you like, we can discuss after the talk. But I want to draw your attention to some of the labels. So it might be quite small to see. So you can see there are some triangle, black triangle, white triangle. They are pointing at certain black lines and white lines. So those lines represent collagen fibers running within the rib as well as running from the rib towards the uncinate processes. So we can actually find this direct contact maybe better on the crocodilian side. So if we look at the right-hand side and panel D over here, then the white triangle on the bottom right-hand side, that shows you more of the complete fibers running all the way from the bottom right-hand side on the rib all the way to the top left-hand side of the uncinate. And somewhere in between, you can still see some white color lines. So those are the collagen fibers connecting the two. So now, in addition to the uncinate scar, then we can have this, what we call histological indicator. Once we have this, we show them to the curators at TRL as well as other museums and say, can we cut open some of your ribs so that we can see whether dinosaur ribs also have this indicator or not? And so we did. We cut, I think, more than 10 ribs. I lost count with how many sections we made. But the important thing is we actually found a lot of what we call the high quantity or coarse collagen fibers running from the rib tissues all the way towards the uncinate tissues. Of course, in dinosaurs, we don't always have the uncinate processes preserved. So we over here, we can look at one example. Right? We can look at the unknown Tyrannosaurid over here, and the section on panel B is showing you all the rib tissues, but on the top, there's a label called the US. That's not our neighbor on the south. That's called uncinate scar. So that's where the uncinate scar is located. And if we zoom in all the way to panel D and panel E, we can see patches of black lines. All those black lines are collagen fibers that has been fossilized along with the ribs. And in terms of quantities, they are even higher compared to the ribs in the modern turkeys as well as the modern crocodilians. So we can confirm these observations in some of the fossil species. And we can, outside of Tyrannosaurus, we can also find the same coarse collagen fibers at the uncinate scar in, say, the Pachyrhinosaurus, which was one species of the horned dinosaurs. So even though the uncinate scars looks like a toothpick indentation, they still have this histological correlate, histological indicator saying that those might likely be attachment site for uncinate processes. But what does this mean? So we need to take a step back and look at the family tree of the entire Archosauria. So we look at we combine all the species relationships into one diagram looking like this. We call this a phylogenetic tree, which basically a family tree showing you the relationship among all the species known. And then we can use computer algorithm to say, if we know we have ossified uncinate scars in some of these Panoraptorans labeled by the pink color, if we know some of these species have the cartilaginous uncinate processes indicated by the presence of uncinate scars in some of those lineages, how likely the last common ancestor of all the dinosaurs and and the last common ancestor of all the dinosaurs would have uncinate processes when they are alive. So we run some kind of algorithm based on maximum parsimonies, maximum likelihoods, as well as Bayesian, and some of those numbers are on those nodes labeled A and D. So those are the statistic results or the computation result coming from likelihood results, likelihood method, as well as Bayesian methods. Even though we only have 19 taxa, and I'm still quite skeptical about this result, we can say with what we have so far, we can infer and say that the last common ancestor of all dinosaurs most likely would have uncinate processes. And we should expect, or we can start with a premises and saying that most dinosaurs would likely have uncinate processes. So after today, you might walk around in the gallery and see if you can find evidence for uncinate processes. A little challenge for you guys. And we can further push it all the way back to the last common ancestor of Archosauria as well. However, the data from Pseudosuchian, the fake crocodiles, they are not as complete as I would like. So I would recommend treat this reconstruction at the node Archosauria with a dose, well, with a boulder of salt. And now this is interesting for scholars studying breathing, because uncinate processes, we know they have muscle attachment, and some studies by Cott and his colleague since 2005 all the way to 2010, they realized, oh, the muscle called the appendicular costalis is attached to the uncinate processes. And during breathing, this muscle is activated. So they must play a role in order to facilitate the costal ventilation in, at least in modern birds, or at least in our neighbor, friendly neighbor, Canada geese. So if we can extrapolate this idea into, say, crocodilian, into other fossil dinosaurs, then this kind of uncinate processes assisted ventilation might have appeared quite early in the evolution of dinosaurs, right? We might even, if this is too much rich for you, we can still say maybe the anatomical capacity for this kind of ventilation mechanism has been established early in the history of dinosaurs, and then later they modify this anatomical configuration for ventilations. Now, this is an unknown and not resolved question, so we might be exploring this venue further down the road. Now, we can kind of spend our entire career just looking at anatomy, and I'm sure some of the audience over here have been spending the entire career just working on anatomy. But I'm a greedy person, so I want to move on to the more mechanical side and really try to understand how the anatomy can actually inform breathing before my funding runs out. So if we want to study how the system functions with what the anatomical information we have at hand, we must also know how ventilation has been studying in the literatures. So since about I think 50s and 60s, 1950 and 1960s, with the invention of mass spectrometer, biologists got really excited because that gave them a way to measure how much air is being pumped into and outside of an animal, assuming the animals are cooperative. So usually they will put the animals in sedations, and then they would put some kind of a cup on top of the nose so that they can measure the volume going in and out of the lung. And sometimes they have to be quite inventive. Like on the top right-hand side, you kind of have to modify what a device used for a human being into a device for a weird-looking flamingo beak. So this has been done. And after that, with the advancement in 3D technologies and X-ray technologies, then scholars also move a step forward and use what we call non-invasive technique. So you have an alligator over here on the left-hand side is in a room resting at peace, and then we shoot some X-ray radiation towards the alligator such that we can visualize the movement of the rib segments during breathings. Right, so we can actually quantify the amount of rotation at individual joints throughout the entire ribcage. And further, more recently, in 2016, I think advanced by Brown universities, then we have a further new technique called the X-ROM, which they can capture the live movements of bone with higher precisions. So even though on the right-hand side, you don't see too much movement in a turkey over here, but they are actually capturing the live movement of breathing when the animal is alive. Right? So they have to do some surgical intervention to place some markers along the bone, but this is what technologies has enabled us to do. So we might be thinking, okay, maybe we can find some other non-invasive techniques, right? Because if we use X-ray, it's not really good for the welfare of the animals, right? And for the evolutionary biologists, right, it's also quite difficult to convince a dinosaur to breathe again when they are mounted in a gallery, right? You can try with all the dinosaurs in the gallery. I bet you won't be able to do it. So maybe we can find other techniques. So what the technique I use for my PhD is called the 3D modeling. So we can have individual pieces of bone and use one of these technologies in order to convert a physical specimen, like the pointer over here, into a digital version that we can manipulate in computer virtual spaces. So on the left-hand side, we have this, what we call the light reflective scanners. So it sends out light patterns so that the scanner will be able to know the distance from the camera to the specimens at various angles, and then based on that, the computer would do fancy algorithm to reconstruct the models like this. So that's the ostrich rib. And if you have a fancy camera, you can also do what we call the photogrammetry, which is labeled in the, which is demonstrated in the middle column over here. So you take a bunch of photographs, and then the algorithm would base on the overlapping of pixels on your photos, and they will triangulate the spatial locations for all the dots or the vertices, and then they can recreate some kind of models looking like these blocks of ammonite. Now, quite recently, I think within the past 2 or 3 years, then you have a new technologies. This is a software called Polycam that you can install in your cell phone. You can just open that app, and then walk around with a specimen, and then you can have some kind of 3D models that you can play with. Right? I haven't tried Polycam yet, but if you're happy, feel free to download the app and then just see what you can capture and create. You might be surprised by your innovations. So with all these methods, we managed to create 2 models. It's quite a time-consuming process. So on the left-hand side, we have created an ostrich ribcage. So we scan individual bones, and then we virtually articulate them or put them back in place in a software we call Maya. And then after we articulate in 3D, then we create a volume. This orange-looking blob is created artificially within Maya, such that we can measure the volume of this blob, and then we can apply the volumetric changes during breathings based on the studies from the 50s and 60s, right? So that we can know how much air is actually being pumped in and out. Based on how much air is being pumped in and out, we can see based on that, and then we can infer how much rotation should take place at various joints. And we did the same thing with the crocodilian ones. And this is the kind of the results, right? So on the top, you have the rough estimation, what we call the osteological range of motion, in which we will rotate the rib segments around the joint in Maya to a degree that the bones would almost become disarticulated. So this version on the top is more exaggerated compared to the real-life versions. And then based on the data from the mass spectrometer in the 50s and 60s, then we can say, well, that's too much. But how much should we scale it back down? We realize we need to scale it all the way down to between 30% and 39%, less than half of the range of motion compared to the exaggerated versions of the roll-around. We call them the plausible or osteological range of motion. So this, the version on the bottom should represent a more realistic breathing motion when the ostrich is alive. But scientists like graph, and we like to have numbers to represent our results. So that's the right-hand side figures over here. So VRS, RS, and R, so they represent different ways of breathing, right? We can breathe with just the rib segments, which is at the bottom row. Then we can generate one version of rotations of among the rib segments. And then we can allow the rib as well as the sternum, the chest bone, to move. And then we can generate another version of breathing. And then we can allow the entire rib cage to move, which is plausible even in human. Right? So the entire backbone can be flexed, or the rib segments can be rotated, as well as the sternum can move around. So from the top to the bottom, then we are seeing a gradual reduction in terms of rib cage movement. And we can see the x-axis for each figure represents each joint along the backbone. So we have C19, C20, so that represents the location along the vertebral column. And the semi-transparent orange column, so those represent the maximum rotation at a specific joint. Right? So if we look at the C19 over here on the top row, then under the VRS version, then we have the semi-transparent orange column representing the osteological range of motion, and after scaling, it only has the solid orange column, much smaller compared to the rough estimation based on OROM. So this is one way that we can refine our initial estimations. Another way we can look at it is that by looking at one joint at a time, then we can actually plot the movement of the rotations during breathings. Right? So over here we have a panel, we have a figure on the right-hand side. We can focus on just panel A for our purpose. Over here we can, from the axis starting from 0 all the way to the right-hand side, that's one cycle of breathing. And then we can see we can follow the red color line. So it kind of increased. That means the degrees of rotation is increasing all the way to the top of the tip. That's the end of breathing in. And then they will breathe out, and then the rotation would slowly reduce all the way back to 0. And we can visualize the joint movement in this way. And we also realize if we apply and assume accelerations, meaning that when we breathe in, the rib segments accelerated, and then once they are close to maximum inhalations, then they will slow down, going through a deceleration processes. Then we can have a more curved version of the trajectories on our graph. So that's the dotted lines. And if we compare our estimation with the live monitoring using XROM technique, we realize, okay, the results are not exactly the same. So our model is incorrect in some way. But we can still capture a similar pattern comparable to those seen in XROM techniques. So we are approximating the pattern seen in live animals, even though we cannot capture the precise pattern as in XROMs. So if we apply this in dinosaurs, we might approximate how the movement of the rib cages when the dinosaurs are alive, but we are not able to recreate the exact movements of each breathing cycles in fossil dinosaurs. Very sad. We still need a time machine for that. Okay, after this, then the next question might be, okay, you have this movement of the rib cage to draw air in and out, but how does this movement become possible? They must be driven by some muscles, right? And before we can demonstrate our results, we need to look at how muscle functions has been studied in the literatures. We will focus just on ventilations. So if we look at the literatures, if we trace all the way back to 1846, then we have some studies done on humans, mostly done by medical doctors, I believe, and they want to understand how human would breathe, and then they might want to use that to detect any pathological situations. So overall, since 1846 all the way to Zimmer in 1935, all the way to, say, Tickle in 2007, most of these studies on ventilations in terms of how muscles work, they focus almost always on one portion of the rib cage rather than the entire rib cage. and they oftentimes would need to simplify the geometries. So the rib segments in their model, they are not curved, or not round or curved, and they are usually simplified into a single rod so that they can compute for analysis. So these are some limitations in my opinions. So we thought maybe we can use our 3D modeling techniques and then capture the natural curvatures of the bone so that we can represent the muscles in a more accurate fashion. Right? In order to know how the muscles are organized, we need to do some dissections, like get our hands dirty. So we chose a flying common ravens. We chose a running emu from Saskatchewan, by the way. And also we chose a swimming caiman. So these are our model species to represent all the muscle configurations in archosaurs, in dinosaurs and their kins. So after dissections, we did some kind of dissections like this. So it's not gross, it's in gray color. When we finish this dissection, we are able to map the attachment site of all the ribcage muscles on individual bones. So we can see all these color labels on the left panel over here. So all those represent attachment sites of ribcage muscles. And with this, it's still not enough. We still need to simplify this a bit more. We need to establish what we call a joint system. So in 3D spaces, rotation of the joint is not the same as our rotations, such as I'm rotating my arms here. It's more natural and undefined. Less defined. In 3D modeling, then we need to define a free perpendicular orthogonal axis in order to describe the rotations of the ribs during breathings. So we establish a joint system looking like this so that we can approximate the natural anatomical movements of a rib bones. And then we need to transform some of our muscle data into vector data because the ribcage muscles, they span along the entire rib. Right? So it's not represented by one single tendon, by one single muscle attachment. So we need to separate the entire muscle into multiple units of vectors representing muscle functions at various locations. Now, after this, we are ready. So we can grab everything we have done in Maya, all those estimated movements, the OROM and the plausible OROM on the bottom, grab all the units, like the sizes of the animals, in this case, the size of the ostrich, and then add on to the muscle vectors, add on to the joint system, and then put them in a software we call SIM. Now, you can do the same in an open source software called OpenSim. You can do it in Anybody, a software called Anybody. So there are multiple options for you to do this. And in SIM, we can visualize the muscle vectors as in these red lines. So one line represents one muscle unit. And as the animal breathes, the orientation and the location of that vector would change. And therefore, we can estimate the effect of that muscle in this way. Now, before I can show you the result, we will focus more on moment arms. A moment arm is basically the distance, the shortest distance from the joint all the way to the muscle vectors. So over here on the left-hand side, we have a muscle called the Briardi, I think it's brachialis. And then on the bottom left, you have the JC, that's the elbow joint of the human. And then the blue line represents the moment arm. The moment arm is often used to estimate how easy the muscle can rotate a certain element relative to the joint. And in computation, both moment arm and muscle forces are vectors, so they have magnitude as well as direction. But because we are working with 3D and we don't really have a way to estimate muscle forces in dinosaurs, we don't have the muscle, simply don't have the muscles. But we can transform this formula into the bottom one so that we can assign all the vector, the directional value to the moment arm so that we can look at the moment arm and then determine if this muscle is activated, whether it will rotate the bone to facilitate breathing in, or whether it will rotate the bone to facilitate breathing out. That becomes a way for us to estimate muscle functions. So this is our results. So we have, on the left-hand side, we have the same graph from the OROM and the plausible OROM estimation, right? So we have how much the rotation can take place during ventilations at various costal joints between the vertebral column and the rib segments. On the right-hand side, the right panel, it's showing you the changes of momentum as the ostrich breathe air in and breathe air out, right? So if the rotation on the motion on the left-hand side is positive, and the rotation and the momentum sign on the right-hand side figure is also positive, that means if the muscle is activated, it must generate a torque that will rotate the rib segment to facilitate breathing in, right? So we can assign muscle functions in this way. And we can tie this back to uncinate processes at the very end and say this APC represents a particular crocodiles muscles attached to the uncinate processes. And based on our model estimation, looking at the sign of the momentum combined with the plausible osteological range of motion, we realize for most of the muscles attached to the uncinate processes, they are facilitating inspiration to draw air into the body cavity. And this echoes the discovery by Cott et al. In 2005 as well as in 2010, saying that uncinate processes are likely structures to facilitate inspiration for drawing in air rather than to pump air out. Okay, so not homework. take home message for today is that if you like, you can apply the transitional criteria in the gallery and see if you can identify which vertebrae are from the neck, which vertebrae are from the chest, and which vertebrae are the maybe regions. Right? And you can also see if you can find any signs of uncinate scars as you browse the specimens in the gallery. Right? Not just after you appreciate all the head elements, like the skulls are cool, but spend some time with the ribs as well. They do deserve some attentions. If you like, you can also apply the 3D modeling techniques at home to capture some small elements. You can get your Thanksgiving turkey this year and then use your polycam to capture them and create your own version of the ribcage. And then you can create the same OROM and plausible OROM like I did. And lastly, I would propose that we can use muscle momentum to really estimate the function of certain muscles in both extant living animals as well as fossil dinosaurs. This is still waiting to be explored. We have more way to do simulations, but I think we can end it here. And lastly, I would like to, this long project is not possible without all the support from across the globe. And I just wanted to say thank you to all the lab members at the University of Alberta, all the people that have been hosted me during my collection visit, including today. Thank you, Ilaria. And also thank you for your time.